Okay, so welcome back. You're looking at the third review, sort of, I guess, YouTube video or whatever we want to call this thing, for Bio 211, and we're looking at chapters 4, 5, and 12. And this section is going to focus primarily on chapter 12. What we're looking at here is how do we introduce the purpose of a single cell inside of the nervous system? And then what is the purpose of the cells that support that? What are glial cells? And I don't know if you've noticed, but all throughout my lectures, what I've been doing is incrementally adding more detail to each of the neuron structures that I've drawn for you. So if we started off with one neuron with essentially no detail to it, just identifying the parts of the neuron, what I'm expecting you've been able to do is follow along and add to the complexity. And that's what we're going to build on here. Making sure you pay attention to, again, what makes an excitable cell excitable. It's the movement of ions at the membrane of the cell. Okay, that's what's important. Please don't think that there's information, electrical information traveling on the inside of the cell. There are some cells that do that, but not our neurons. There's transport inside of a neuron. If you read closely, there's like slow axonal transport and fast axonal transport, but that's not what we're talking about. We're talking about a cell being excitable. Those transport mechanisms are for moving something like neurotransmitters to the end, the axon terminals of a neuron. So the purpose of a neuron, of course, is to actually send electrical information in in a unidirectional way. Okay? Think about the fact every time we've sent electrical information, it has moved in a specific manner, and that's gone from, say, this line represents now the cell body, this line represents the axon itself, and this is the axon terminal. When we think about the graded potentials that are summed here at the axon hillock, they become action potentials that travel along the axon, and then this causes the release of neurotransmitter because voltage-gated calcium channels allowed that to happen. Now think about how simple I drew this cartoon, because really this superimposes on top of this cartoon here, right? Graded type of potential here is excitatory. It's summed here so we could reach that minus 55 threshold, and then because it traveled the length of the axon terminals, opening up a specific subset of channels here, we can now get neurotransmitter release into a synaptic cleft, NT. Okay? So let's get rid of that. Okay. And once we've gotten rid of that, what I want to make sure we do is you understand what I've done here is it's different than, say, a glial cell. Glial cells are regulating the function, function, of nervous system by supporting the neurons, N-E-U-R-O-N-S. And remember, there are different types. We have astrocytes, we have microglia, IA, as well as microglia, and ependymal cells, ependymal cells. So again, you got those astrocytes, microglia, and ependymal cells, but they're not the only glial cells. You could actually have a Schwann cell in there, Schwann cell, or oligodendrocyte. I'm going to put that in double brackets, oligodendrocyte, C-Y-T-E. Okay, I think that's an E, oligodendrocyte. No, who knows, whatever. Okay, so the significance here is this. And I have to admit, I assumed this when I wrote the question. You guys have figured out that the nervous system itself has a central nervous system component to it, and it has a peripheral nervous system component to it, right? Within each of these, your entire nervous system can be broken down into voluntary, vol, which is going to be your somatic, okay, and you have involuntary, VOL, which is your autonomic. And we spend a whole lot of time inside chapter 15 breaking down the autonomic nervous system. The somatic is what we've spent essentially most of our time talking about. And I don't want to spend too much time separating this out for you right now, but in your review, make sure you can think about the human body in terms of here's the spinal cord and the brain, and that's our central nervous system. The nerves that travel into it and out of it, these are all part of your peripheral nervous system, right? For your lab practical, you have 31 pair of nerves. You have 12 cranial nerves. Remember the cranial nerves. I want to make sure you guys know where the nuclei for these things are. Nuke. You'll see why that's really important when we get inside chapter 15, but that's NUC for the cranial nerves. And that's what I'm going to be testing you on, because the significance here is that's what regulates normal physiology. Okay, so having reminded you of all of that, what are these glial cells? What are they going to do? So what does the astrocyte do? What does a microglial cell do? What are these Schwann cells and oligodendrocytes doing? Well, these create myelin that is then going to surround the axons of neurons. Astrocytes create physical structures for separation, for diffusion to occur. Microglia are essentially white blood cells that are running around the nervous system inside the central nervous system to make sure that nothing, you limit the amount of infection that occurs inside there and removes the debris. Okay? So there's a lot going on in this little slide here. Now that you've figured out all of this stuff, how does a stimulus become electrical information? Well, I want to get rid of this cartoon, redraw this cartoon here. Notice one. Actually, let's just draw the actual multipolar neuron to make sure everybody's on the same page. This is a multipolar neuron, right? Right, where we're now going to put in the axon. Axon is here, cell body is here, axon terminal is here. And these little regions right here are, of course, let's say we are inside the peripheral nervous system. So these are all Schwann cells, and they're myelinating this neuron. So a stimulus, by the time you finish chapter 12, you know there are multiple types of stimuli. You can have a chemical stimulus. You can actually have some sort of pressure stimulus. And you'll notice I use the skin to review all of these, right? Free nerve endings feel ions or changes in ion concentration, right? When you actually crush them. Pressure, these are usually attached to, say, corpuscles, where the free nerve ending is inside of that. But all of this information is sent back to the cell body, which then travels as a potential. And the potential comes in two flavors, right? It can be depolarizing or hyperpolarizing. But the important part here is you're studying how it is we count the depolarizations and hyperpolarizations, because what I'm definitely going to go over on Monday, but I want to make sure you guys are paying attention to, is that an action potential is traveling alongside here is a sodium, and I should say voltage gated, so I'm going to go VG, voltage gated sodium channel, and a voltage gated VG potassium channel. And this is what I'm talking about when I'm saying electrical information travels this length here. Down here at the synaptic end bulbs, we now populate them with voltage gated. Notice I'm describing the type of channel here. You can't ignore that. Voltage gated calcium channels, Ca2+. That's why we need calcium so much. Okay, to allow for the release of neurotransmitters. Great. Okay. So you get the general idea. Let's think about what's going to happen next, though. Okay? How does this interpret? The interpretation is, of course, occurring, one, you could say here at the cell body, is it excitatory, is it inhibitory? If we reach threshold, which is minus 55 millivolts, right? Minus 55 is the magic number, MV. To get us from minus 70 to minus 55 causes the all or nothing, which then will cause the release of that neurotransmitter. There's no question about that. Okay? Let's erase this and now think about how do you actually create that neurotransmitter. NT equals chemical information. A neurotransmitter is simply a chemical. So when we go over those in class, I'm hoping you've already read about them. You're thinking to yourself, oh, he didn't talk about that. I'm not responsible. Remember, you had to read chapter 12. I'm expecting you've read these things by the time I see you tomorrow. Okay? Let's get rid of that drawing. Let's move on to the next figure. Okay? So draw a neuron, draw a glial cell in the peripheral nervous system. I already did that. Okay? And you're going to see that what I want you to do here, just like I did in the previous question, is can you do this? So say, if it's in the peripheral nervous system, do you understand? Let's see, draw this out here. Let's make it simpler. Cell body, axon, axon terminal. Can you decorate this with all of the different channels that are responsible to function here? I mean, all of them. Are we talking leakage channels? Are we talking voltage-gated channels? What else do you need to know that's on these, right? And where are they and what are they doing? So not only do we have leakage channels, voltage-gated channels, what about the ionotropic channels here, or metabotropic? Notice I put them in a specific place on this representation of a multipolar neuron. Okay? Notice this is pretty simple. And what I mean by that is draw these three lines. Make sure you know what's going on in each part of these. Make sure you understand this represents the axon hillock right here, right? So you've summed all of these so you can actually go through the actual steps of an action potential. Notice the action potential is only happening on the axon and the axon terminal. It doesn't happen over here. Okay, so pay attention to those really simple differences. With all the physiology you've learned, explain figure 1210 inside your text. And I'm without a textbook in front of me, but what I think, if I remember when I was writing this question out, this is that first order neuron that activates the second order neuron that travels to the thalamus that ended up traveling to the cortex of the brain. And then you have an upper motor neuron that activates a lower motor neuron. And notice there's a directionality here. I'm going to put these things in here so you figure it out. Okay? And you'll notice that there's myelinated structures and unmyelinated structures, and we change the thickness of the axon terminals and cells. But notice when I'm doing this, axon terminals are facing a specific direction, right? What does that mean when you're looking at that student with their pen in their hand? Notice the end of the pen activates sensory receptors while they're writing, horrible things about their instructor on a piece of paper while they're studying. What is actually going on here? Notice that these are A-type fibers because they're thick axons with lots of myelination. But these interneurons here, okay, these guys, I should say, axons without myelination would be interneurons, and that would mean that they would actually have slower transmission of information. These guys down here, what is the difference between an A-type fiber, A, B, and C? And notice all interneurons, I'm going to put the I for interneuron there, are all C-type fibers. All somatic sensory, SS, and somatic motor, SM, are all A-type fibers. Things get a bit twitchy here when we actually get to B-type fibers. We spend a lot of time thinking about them later, but inside lab, you know what this looks like. Myelinated are white portions of the brain and the spinal column. Unmyelinated are gray portions, okay? And that would mean that all those gray portions are made up of interneurons in the cell bodies or dendrites of other myelinated structures, and you'll see why that's significant. Okay, so cool. Let's get out of there. Last couple of minutes here, compare and contrast graded and action potentials. Now this is a really important question, and what I mean by that is, what's the difference between, come on Jonathan, what is the difference between this and this versus, remember, this is what's being summed till we get to here and here. And really, I want to make sure you understand that This is occurring because we're summing all of these at the axon hillock. It's only if we hit that minus 55 that we actually have an action potential, right? So if we just draw a quick multipolar neuron for you here, we're running out of time. That quick multipolar neuron, put some dendrites on it. Okay. We sum up all of these across the membrane, right, at the axon hillock here. If we actually get that, then we have these traveling down here to open up our voltage-gated calcium channels, which will then let the neurotransmitter into the space. And that space is, of course, called the synaptic cleft. Now put two of them back to back. Okay? Put two of these neurons back to back and explain what actually happens. Cool. Okay. Shut that off. Let's go here. Last one. How do neurotransmitters work? In the next minute or two, I'm going to break them down for you, right? So they're all different sized. So we have different types of neurotransmitters, TYPES of NTs, right? Who really cares? For the purpose of this exam, what you want to make sure is you know their categorizations, if they're excitatory, EXC, or inhibitory. And that would mean that they either cause a depolarization, right, which would be excitatory, EXC, okay, or they're inhibitory, inhib, okay, or I should say hyperpolarization, HYPER, and that would mean that they are inhibitory, HINH, inhib, okay? So study that first, and then draw a couple of neurons alongside one another, meaning you have, let's do this quickly, work through the problem, right, where you're drawing a couple of multipolar neurons. I don't care if they're myelinated or unmyelinated. You can practice continuous or saltatory conduction. Nudge, nudge, hint, hint, wink, wink. But I want to see what happens if we actually have an inhibitory neurotransmitter being used here, right? What would happen to this neuron there? Think that through, because that's really important. If you don't understand that the chemical conduction of signaling here can stop, well, then what's the point of this class? Okay? Okay, enough for now. Go back, make sure you read these in terms of, like, are they excitatory or they're inhibitory? And we'll go through the important ones, namely acetylcholine, ACH, okay, versus, say, I don't know, a glutamine or something like that, GLUT, okay? Okay, enough for now. Have a good night. Hopefully you listen to this and you're ready for class tomorrow.